This is the Master Brewers Podcast, brought to you by the Master Brewers Association of the Americas, a volunteer organization dedicated to continually improving the products and processes of our membership since 1887.
Let's go!
Master Brewers brings you interviews with the industry's best and brightest in brewing science, technology, and operations.
Calm down, I'm moving too fast.
This Master Brewers podcast is proudly sponsored by Barnett Mechanical, a full-service design build firm specializing in turnkey process and utility systems for the brewing industry. You know beer. We know breweries.
Is it safe to assume that what might work in a hospital to sanitize, an operating room, was that equally effective at sanitizing my cellar? And through these experiments, I confirmed that no, I don't believe that is a safe assumption. So I think we as an industry have a lot to learn.
This week on the show, more great experiments from Bell's Brewery. This time, we're looking at the efficacy of common brewery sanitizers.
Hi, my name is Elliot Parcells, and I'm with Bell's Brewery.
Hi, I'm Josh Pohlmann. I'm with Bell's Brewery.
What prompted you guys to run experiments on your sanitizers?
Well, it just happened that shortly after I took the job as a shift brewer, we were making a change to our spray bottles out in the cellar. We had been using ethanol for the longest time, and we had just made a switch to iodophor in spray bottles, and that was based on some reading we had done on the web. And it didn't really sit right for a couple of reasons. I've used alcohol in the past and kind of believed in it. And then I recalled from John Palmer's homebrewing book that I remember him citing a 2-minute contact time for iodophor. And so I wasn't sure if that made it an appropriate spray,
Okay.
And so we made the change and started doing it, and it just kind of prompted me to go home and start reading and kind of investigate what's known about these two things, and kind of went down the rabbit hole in the literature and came up, not really sure whether what I was doing out in the cellar was working. And I had a friend, Elliot, in the lab here who was interested in what I was complaining about and telling him what I was learning. And one thing led to another, and we found ourselves involved in some experiments that taught us a lot.
That's great. It's always good to keep challenging, what you think you know. So why don't you start off by telling us, which sanitizers do you guys use at Bell's and why?
So we use a variety of sanitizers at Bell's for different purposes. Looking at for something that's quick, a very quick contact time, we like to use ethanol. We found that most alcohols behave the same way. We still use iodophor for parts buckets, and
we'll spin our rack rigs with it.
Yeah, and peracetic acid as well.
Yeah.
For tanks and some rack rigs as well.
Yeah.
And for this study, you primarily looked at ethanol versus iodophor. How about walking us through sort of the pros and cons of each of those?
Sure.
So, yeah, absolutely. Ethanol, rapidly bactericidal. The mechanism of it working is pretty well known. It just denatures proteins and penetrates cells very quickly. There are a couple cons to it. One is it evaporates very quickly, especially in high concentrations. If it's too concentrated, if it's 100% ethanol, it's actually been cited to be not as effective. It also has no sporicidal properties. As far as iodophor goes, it is bactericidal, it is sporicidal, and there is a slow release of the free iodine, so it's it does degrade, but at a slower rate than ethanol does. Some of the cons are, though, what we found is there's a relatively long contact time required, and if it's not diluted properly, it is actually quite ineffective, or cited to be quite ineffective. And the mechanism of its kill is, as far as we can tell, is still not fully understood.
Yeah. theory, really.
Yeah.
One thing I like about ethanol as far as spray bottles go is that it's got this property where if you spray inside a butterfly valve with it, it coats the inner surface of that really well, whereas if you have a water-based thing like iodophor, it kind of beads up, and it always made me question whether or not I'm actually hitting the entire surface like I want.
What was your initial hypothesis here when you got started with these experiments?
I was just going to put my faith in Pohlmann's statement that the iodophor would have a 2-minute contact time for organisms that are of interest to brewers. And I also thought that I had a hunch that alcohol might sanitize a little faster, but I wasn't clear how long it would take.
Yeah, I was looking at kind of the manufacturer's recommendations and saying, at the working concentration, basically that they will perform as advertised or better.
So tell us about your first experiment.
So I was actually in the middle of a microbiology class at the time, and we adapted it from basic micro 101 course where we would take a number of sanitizers, create them freshly prepared at working concentration, and then we would take a number of organisms and we would inoculate these sanitizers with the organism, and after a certain amount of time we would then do plate streaks, so just a simple streak on Petri dishes. And those Petri dishes were divided up into 4 quadrants. So we had a control streak where the microbes were not exposed to any sanitizer. And then in each quadrant, it was associated with a time frame. So our first experiment was relatively quick. We let the organisms sit for 15, 45, and 90 seconds. So here we're looking at that up to this 2-minute contact time, but not quite. So 90 seconds was something we thought, it wasn't quite arbitrary. We were thinking, as a shift brewer, we're out on the floor. I don't have time necessarily to, spray a perlick, spray a connection, and wait for 2 minutes all the time. We have other things to do. We have beer to move and all that kind of stuff.
There's probably going to be somebody yelling at you if they see you standing there all the time for 2 minutes at a time, right?
Right.
Sure, exactly.
What are you doing right now? I'm just waiting for contact time.
Right. And so we were kind of thinking, okay, 90 seconds seems, on the far end to be reasonable. And that's, pretty simple design for that experiment, and just wanted to see what would happen.
Did you mention which bugs we used?
Oh, no. So, yeah, we chose, so first of all, we chose lactobacillus. So this is, pretty obvious one in the brewing industry, doesn't need much explanation. Brettanomyces, it's another one, wild yeast. And part of the reason why we chose these 2 organisms in particular was one is a prokaryote and one is a eukaryote. So what we found is, a lot of these sanitizers, they have, different requirements for different organisms. So we really wanted to get a good genetic spectrum. So we also chose a brewing yeast, our Belgian yeast, and also a species of Saccharomyces diastaticus as well, which has been in the news quite a bit and can be quite a threat for breweries.
Yeah.
Walk us through those results. What'd you see?
Yeah, so we found some. It's pretty interesting. So for each of the concentrations, so we chose for alcohols, we chose isopropanol and ethanol.
When you say ethanol, are you talking about like Everclear, or what do you mean?
We're talking about laboratory grade ethanol.
All right.
So although we didn't use Everclear, but I suspect it would have very similar results. That's an interesting one. So we chose 100% ethanol and a 70% dilution as well. What we found is between the two, there was no difference. Within 15 seconds, none of the organisms showed any growth. But what we found for iodophor, which is interesting, and here we were titrating, freshly prepared up to 25 ppm, which is the recommended concentration to be considered a no-rinse sanitizer by the FDA.
Right.
And what we found growth at 15, 45, and 90 seconds. So we were able to overwhelm the sanitizer
Wow.
in this particular case.
That was the big surprise to me.
Yeah, we were surprised to say the least.
Coming up,
different microorganisms have different resilience to different chemicals as well. We can adjust our contact time for the worst case scenario.
I'm John Bryce, and you're listening to the Master Brewers Podcast from the Master Brewers Association of the Americas. This episode is brought to you by
ABS Commercial is a full-service brewery and parts outfitter. From our Raleigh headquarters to our Denver office, we proudly offer brew houses and fermenters from 3 barrels and up, yeast brinks, boilers, kegs, chillers, tri-clamp, and other stainless parts, all with the quickest delivery and lead times in the industry. Learn more at abs-commercial.com or call 877-BREW-ABS. ABS Commercial. We are brewers.
Here's what's coming up on the Master Brewers calendar. District Western New York meets at Railhead Brewing July 11th. District Northern California meets at Admiral Maltings in Alameda July 19th. District Pittsburgh tours City Brewing in Latrobe July 21st. Don't miss the brewery safety webinar July 25th. District St. Paul Minneapolis joins forces again with the Minnesota Craft Brewers Guild for the 5th Annual Business and Technical Conference July 27th in Duluth. The District Milwaukee Summer Picnic is July 28th at Capitol Brewery in Middleton. The Diastaticus Detection Methods and Control Measures webinar is August 1st. District Philly holds its summer meeting at the Pennsylvania Sam Adams Brewery August 3rd. The annual District Texas summer meeting is the weekend of August 3rd in Kerrville. The ASBC MBAA Brewing Summit takes place in San Diego this August. Register at mbaa.com, where you can also view the full calendar of events with more details or find a district meeting near you. Now back to the show.
Now, for this particular experiment, we didn't do quantification. So we didn't quantify in CFUs per mil what survived and what didn't. But what we did find was pretty interesting. If you looking at some of these pictures, you can see throughout time there is less growth on the plates.
Yeah, and I wish listeners could see the photos from your presentation, and I'll mention where they can be found at the end of the episode. But why don't you explain your method for streaking and inoculation a little bit more, visually, because I think it's pretty cool.
Sure. So what we do is we grew these microorganisms up to do a very healthy concentration, something you'd see like in a propagation. So they're very, very concentrated. If you were to encounter them in this concentration in your brewery, it would be worst case scenario. And so we had 9 milliliters of the freshly prepared sanitizer, and using a pipette, sterile pipette, sterile technique, we moved 1 milliliter of organism into that tube of sanitizer. And then with just a loop, we would take, invert the sample, take our loop, and do just a single line in each quadrant. And we were looking for go, no go. At Bell's and most, imagine most breweries, we have a zero tolerance for these organisms.
So the big take home from that first round was just that the iodophore hadn't killed any of those 4 bugs within a 90-second window. So in round 2 of our experiments, we extended that window out to about 8 minutes, and we found the same result. So we still don't know how long it takes iodophore to kill, because we've only gone to an 8-minute window.
At 25.
At 25 parts per million. I apologize.
And yeah. What was, as you went to set up the second experiment, sort of what was your hypothesis there? What did you set out to do kind of in your round 2?
I think at that time when we were about to get into round 2, I was still thinking, we only did a 90-second window, and I was kind of predicting it would work within 2 minutes. So I kind of stuck with my 2-minute hypothesis that this would work. So I was particularly shocked when it continued to, not clear all the bugs out of solution out to 8 minutes. That was at 25 parts per million. If we increase the concentration of the IO from 25 up to 50 or 75, it did kill faster, and we were able to get clean plates in the 2-minute window, right?
Yeah. So our goal for round 2 is now we just want to see it work. So we are going to up contact time, and we are going to up concentration as well to try and find the correct window.
And what was that window?
We were able to show that iodophor at certain concentrations is effective. So at 50 ppm for lactobacillus, we did see growth at 2 minutes, but after 5 minutes, we saw no more growth of microorganisms at 50 ppm. At 75 ppm, we saw no growth even after 2 minutes. So we were able to correlate concentration and contact time to efficacy here. What's interesting, and I really like this part of the results, is for the Saccharomyces diastaticus, for the eukaryote, it actually took longer for iodophor to affect these organisms. It took between 5 and 8 minutes. So we saw growth at 5 minutes, but none at 8 minutes. So not only were we able to correlate kind of this known concentration, contact time, and sort of show that in a repeatable way, but we were also able to show that different microorganisms have different resilience to different chemicals as well, and we don't fully understand the implications of that. But what we do know is we can adjust our contact time for the worst case scenario.
Has this resulted in any kind of major SOP changes at the brewery?
Yeah, and there's probably a few more to do in the future, but based on the results from those first 2 rounds of experiments, we took the iodophor spray bottles and swapped them back out for ethanol spray bottles right away. The one thing that's the jury's out for us is we still do use iodophor to spin our rack rigs, and we tend to spin for about 20 minutes, and because we've only explored out to an 8-minute contact time, we're still not sure if a 20-minute window, a 20-minute spin with iodophor is sufficient to call your rack rig sanitized. I'd like to think so, but I haven't proven that yet.
Right.
We also, so in the second experiment, we did focus on ethanol and iodophor. There's a lot of interest in peracetic acid as well. Peracetic acid seems to be more effective than iodophor. We don't know for sure. We haven't carried out those experiments yet, but we do tend to lean towards peracetic acid for some of the processes where there's more risk.
Yeah, okay, that makes sense. And it seems like a lot of the industry has transitioned to peracetic, away from iodophor over many years. So I guess, is that what's next for you guys, is kind of really digging more into peracetic acid?
I think so, yeah. There's a lot to learn still on peracetic, still a lot to learn about iodophor as well.
Yeah, we're also interested in some stability as well. So one of the reasons why people like iodophor is it does hold, it is fairly stable. We do know that it degrades, but we don't know exactly how quickly it degrades and how that compares to peracetic acid. One of the lingering questions I think that we have as well is, once you've sanitized a loop or a tank, it is always best practice to sanitize and use it. Exactly.
Right.
But that's not always the case in a production setting. Something can happen. And the question that is always in people's minds is, what is this window before we have to resanitize? So
if you spin a tank, how long? What's your window before you got to respin it? If you spin a rack rig, how long can it stay packed and be considered sanitized before you got to spin it again? These things all depend on the stability of these sanitizers, and we have very little experience with how stable they are. I took 1 bucket of iodophor, I think it was at 25 parts per million, and I just titrated it over the course of almost 2 days, about a day and a half, and it's the parts per million of active ingredient cut in half over that time. To me, that's pretty stable.
Yeah.
Obviously you'd want to use your rig fast, but if it's got to sit packed, it looks like you've got some tolerance there.
Yeah, and I'm sure storage conditions are a factor there, as are some other stuff too. I thought it was really cool that you also included photos that showed the color of iodophore at 2550 and 75 ppm. I mean, 75 is pretty dark, isn't it?
Yeah, it's right at that point where, if you're taking really strong iodophore and you're slowly adding water, it's initially very cloudy. You have that like metallic cloud to it. And as it makes that transition from being cloudy to clear, where you can actually see your brush, the bottom of the bucket, that's about 75 parts per million. So that's one of the nice practical advantages to using iodophore, is you can kind of visually titrate it roughly.
Right. And so, that is another, exactly. That's, people love iodophore for that reason as well. We still, and definitely stand by, we need to titrate our chemical before we use it, because we need to validate that we're reaching, the correct concentration, because otherwise then the contact time changes drastically. So
I think one of the biggest take homes for me in this whole experience was that when this thing came up in the brewery where we were playing with our sanitizers, and I went home and dug into the literature, I feel like a large majority of what we understand in the brewing industry about the sanitizers we use is based on data that's derived from the medical industry and human pathogens. And so one of the big aha moments for me was to ask the question, is it safe to assume that what might work in a hospital to sanitize an operating room, was that equally effective at sanitizing my cellar, and in those critical areas? And through these experiments, I confirmed that no, I don't believe that is a safe assumption. So I think we as an industry have a lot to learn, to be sure about what we're doing with these sanitizers. And I'm happy to have gotten a glimpse into that now.
Yeah, and I would like to add that I think this experiment's quite easy to carry out and follow. And so definitely recommend that anyone who's interested in this, and especially other sanitizers that we haven't covered or haven't gotten to go ahead and give it a shot and find out what works for you. Whether taking into account contact time, cost, all of these things play into what chemicals you purchase. But for peace of mind, I think this is a nice experiment to run.
That was Josh Pohlmann and Elliot Parcells here on the Master Brewers podcast. This work was presented during the 2017 Master Brewers Conference in Atlanta, which means you can view the slides with all the great pictures when you download the conference proceedings from mbaa.com/store. I'm really looking forward to the ASBC MBAA Brewing Summit coming up this August in San Diego. It only happens every 4 years, and it's not like any other conference you've attended. The Brewing Summit is 100% the science and technology of brewing. No pep rallies or business lectures, and you'll be surrounded by some of the smartest men and women in our industry. If you can only attend one conference in 2018, this should be it. Register now at mbaa.com.
When we came around there,